Hey, welcome back, everyone. Hopefully that previous exam went well. If it didn't, come see me. We can go over some stuff, get you pointed in the right direction. We are past the halfway point. We're approaching the home stretch at this point, and we're getting ready for that cumulative final. But we still have a couple more exams to go before we get there. So today we're talking about photosynthesis, which in many regards is the exact opposite of cellular respiration. Like if you look at the chemical reaction for the two, you just flip one and you've got the other. The only difference is for photosynthesis, your energy input is light. But the other major difference is that cellular respiration takes place in mitochondria, and photosynthesis takes place in chloroplasts. Okay, both plants and animals carry out cellular respiration, which breaks down glucose, utilizes oxygen, gives off carbon dioxide, takes place in mitochondria. Plants, algae, and cyanobacteria photosynthesize, animals do not. This occurs in the chloroplast, builds glucose, utilizes CO2, and gives off oxygen. The nice thing about this is that in these photosynthetic organisms, their net is that they do more photosynthesis than they do cellular respiration, so we have plenty of oxygen to breathe. Both utilize the electron transport chain and chemiosmosis for ATP production. Photosynthesis converts solar energy into chemical energy. These organisms are called photoautotrophs, and that's where the vast majority of the chemical energy and biomass on the planet comes from, and then other heterotrophic organisms feed on them. There are some, chemioautotrophic organisms that do weird things like use sulfur and radiation from rocks and just weird stuff like that in order to carry out chemical reactions. Both autotrophs and heterotrophs use organic molecules produced by photosynthesis, which is why ultimately the sun is the source of all life on this planet. Okay, so flowering plants as photosynthesizers. Photosynthesis occurs in the green parts of the plant, and that is because green light is kind of useless for photosynthesis. It's just too low energy. So their pigments absorb everything but the green, and it reflects the green light back, which is why it appears green to us. These tissues are loaded with chloroplasts, so I guess that's where your photosynthesis takes place. And they contain chlorophyll and other pigments that allow them to capture solar energy. So chlorophyll A and B, those are both kind of a greenish color. Your carotenoids are kind of an orangish color, and if I'm remembering correctly, xanthophyll is more of like a reddish brown kind of color. We'll see that in the lab. We'll actually separate these out, and it's kind of a cool experiment. So overview: photosynthesis takes place in the chloroplast. It has a nice little double membrane. Again, the evidence of the endosymbiosis theory, just like mitochondria. As a fluid-filled interior called the stroma, and then you've got these stacks of thylakoids, and then the thylakoids together are called a stack of them together is called a grana, and they are connected to each other. It's a continuous space. They've got these little tunnels in between them, so it's a fairly complicated structure. So again, you've got this photosynthesis reaction. You flip this the other way, oxygen plus glucose. Oxygen plus glucose yields CO2 and water. That would be... Okay, we're going to edit that bit out because reflux sucks. Oxygen plus glucose yields water and CO2. That would be respiration. But going this way, carbon dioxide plus water plus light energy yields glucose and oxygen. And also you get chemical energy in the form of ATP in your cellular respiration. So, they're basically reverse reactions at molecular level, but there's a lot more going on. So you've got waters being taken up by the roots and transported to the leaves by the veins in them, and we'll talk more about how that occurs in a little bit in a following chapter. CO2 enters the leaves through stomata. Again, more detail later. And then light energy is absorbed by chlorophyll and other pigments in the thylakoids of the chloroplasts. You get then you get your glucose and oxygen at the end of that. You have two sets of reactions in photosynthesis. You have your light-dependent and your light-independent reactions. The light-independent reactions are also referred to as the Calvin cycle. The light reactions convert solar energy to chemical energy in the form of ATP and NADP when this releases oxygen. The synthesis reaction of photosynthesis is producing your carbohydrates, your glucose, and that is done using the light-independent reactions or the Calvin cycle. It makes sugar from CO2 fixation. So with that in mind, we'll look at these independently. So the light reactions, and again, this is like the respiration lecture. You need to know where these things occur, what you get out of them, what goes into them, what goes from one to the next, that sort of thing. So you got two pathways in your light reactions that both transform solar energy to chemical energy. One produces ATP, both produce ATP, and then one also produces NADP, and ATP and NADP link the light-dependent and independent reactions. So the light-dependent reactions do have different pigments within the thylakoid membranes that absorb energy on different wavelengths, and they're essentially, the pigments are essentially an antenna for gathering solar energy. Green light is only minimally absorbed and is reflected, so they appear green because there's not a lot of energy in green light. And then other pigments become noticeable in the fall when the chlorophyll breaks down. What that's essentially telling you is that the plants are going dormant and the leaves are dying because they're no longer being used for photosynthesis. Non-cyclic electron pathway. So the pigments complex absorbs solar energy. The electrons become excited and are passed from one pigment to another until it is concentrated in the reaction center using chlorophyll A. So basically, when a photon strikes one of these pigment molecules, an electron gets excited and jumps up into a higher energy orbital, and then you just kind of pass those around until you concentrate them into one particular place where you have sufficient energy to do something with it. And what they end up doing is they excite the electrons in the reaction center, and then those get excited enough to escape the reaction center and move to electron receptors, so now you can start actually moving the electrons from the antenna where you've been collecting solar energy, essentially. Photosystem 2, the electron acceptor receives the energized electrons from the reaction center and then sends those electrons to the electron transport chain. Energy is released is used to pump the protons into the thylakoid space, forming a proton gradient, which then flows through ATP synthesis, and then you use that ATP in the Calvin cycle. So this should look remarkably similar because it is almost identical to the electron transport chain used in cellular respiration. We're just using light as our energy source in order to bump the protons down. But this final step here, where you're passing, or this process here where you're passing electrons, from one carrier to another to produce a proton gradient, and then you collapse that proton gradient through ATP synthase, is essentially the same process. And then you send that off to the Calvin cycle. That's the extra step here. There are a class of chemicals, molecules, however you want to phrase it, that are proton decouplers. And so what they do is they just kind of collapse this proton gradient by allowing the hydrogens to flow out through something other than ATP synthase. Basically just ends up producing a ton of heat. Infants have brown adipose tissue because when they're newborn, they're too small to be able to shiver to produce their own heat, and brown adipose has its own decouplers in it. So basically, you expend a ton of energy building this proton gradient, and then it just collapses, and you build it again and again and again, and so it produces a ton of heat. At one point in time, these decouplers were actually sold as a miracle weight loss drug because, as you can imagine, continually running this process and getting nothing out of it would consume a lot of energy. And it is true, if you are taking these decouplers, you will lose weight, and very rapidly at that. You'll also be, uncomfortably hot like 98% of the time. The problem is that the cells in your eyes, which do not have blood vessels going to them, will rely on the diffusion of ATP in order to carry out their metabolic processes. So if you take these decouplers for too long and you're not producing sufficient quantities of ATP in the surrounding tissues, the cells in your eyes die and you go blind. And if you continue taking them, or have taken them in sufficient quantities, and you notice you've gone blind and you still have enough of it in your system, you basically starve to death at a cellular level, and it can result in death. Though they do still have a function these days, because if you are a Russian Spetsnaz, you have a bottle in your vest that has these decouplers in them. So if you, say, fall through the ice in the middle of the Siberian tundra, or I'm sorry, if you fall through the ice in the middle of Siberia, you can pop one of these pills to produce enough body heat to stave off death by hypothermia. Very risky, but you're kind of hosed anyway, so roll the dice. But the FDA does not permit these to be sold as diet pills. As a matter of fact, I don't think that there's any way you can acquire these legally in the United States anymore, but because of how dangerous they are. But anyway, tangent concluded. Let's move on to the next step here. Photosystem 1 comes next in the non-cycling electron pathway, and it absorbs light energy via a different electron acceptor than photosystem 2, which we just talked about, and then it passes electrons to NADP, which becomes NADPH, and then that's used in the Calvin cycle. So, photosystem 1, photosystem 2 comes first, then photosystem 1, and photosystem 1 is also the one that produces the NADPH, but they both produce ATP. If I'm remembering correctly, they're named in order of their discovery, not the order in which they actually take place in the system, which is why 2 is before 1. Now, your light-independent reactions here, carbon fixation is the first step of the Calvin cycle. And so basically what you're doing is, well, let me just read through this and then I'll summarize it in English. So your CO2 is attached to a 5-carbon RUBP, which gets you this 6-carbon molecule, which you then split into 2 3-carbon molecules, and then there's an enzyme required to do that called RUBP carboxylase, which splits it in half. Then you take those 3 and you 3-carbon, or the 3 PG, and reduce them to G3P in 2 steps, and then the energy and electrons are supplied by using the energy. There's an electron supplied by ATP and NADPH, and it takes 3 turns of the Calvin cycle to allow 1 G3P to exit as glucose. Okay, so what does that mean in English? You've got your 3 CO2 molecules being brought in here, and you are fixing them to a 5-carbon carrier, and you are burning 6 ATP, taking the energy from 6 ATP, which reduces them to ADP, and then you need 6 NADPH, and you are reducing them. And remember, oil rig reduction is gain of electrons, so you're taking 6 electrons and attaching them to it, And so now you've got this your 6 molecules of Ga3P, which has, half a glucose basically. You regenerate your RUBP, because you got back down to 3 molecules here. You lost 1 molecule of Ga3P, and you've got half your glucose, and then you start the process over again. You're going to get 3 ATP out of it here. So each cycle you're using, you burn 3 more ATP here. So each cycle you're using 9 ATP. You do this 3 times, and eventually you end up with a glucose molecule. There are some alternative pathways for photosynthesis, different ways it kind of takes place. The default is your 3-carbon, which is kind of what we've already talked about. And so your C3 plants have a different structure than C4 plants, which we'll talk about in a minute. So C3 plants are things like wheat, rice, oats. They have their mesophyll cells and the leaves of the leaves in parallel layers. And they bundle in sheets around the plant veins and do not contain chloroplasts. As a result, cells using the Calvin cycle are exposed to CO2. So the problem with that is it slows down your Calvin cycle. It inhibits it. I believe this is actually a typo right here, and this should be exposed to oxygen, because as it explains over here, when RUBP carboxylase binds oxygen as well as CO2. When bound to CO2, it undergoes photorespiration, which is a wasteful reaction because it uses uses oxygen and releases CO2, decreasing the output of the Calvin cycle as the oxygen concentration in the leaf rises when weather is hot and dry, because the plants keep the stomata closed to conserve water. So this is actually a typo. The problem here is that the cells of the Calvin cycle are exposed to oxygen, and it decreases its energy output. And the reason the oxygen concentration rises in the leaf when it's hot and dry is because the stomata remain closed to conserve water. During hot, dry weather, the plant is producing less and less energy, and so it either dies of dehydration or lack of energy. C4 plants kind of get around this a little bit. They have a slightly different process. So C4 plants are things like sugarcane, corn. The mesophylls are arranged in concentric rings around the bundle sheath, and they do contain chloroplasts. And in the mesophyll cells, CO2 is initially fixed to a 4-carbon molecule. So there's this extra little step right here, which is then later broken down into a 3-carbon molecule in CO2, and then the CO2 enters the Calvin cycle. So the nice thing about this is you're not dumping oxygen where you're trying to do your Calvin cycle. You have this extra step that kind of physically separates the two. And that means that they can continue functioning in a hot, dry climate because they're not being choked out by the oxygen buildup. You are still building up oxygen in the leaves, but that's okay because you're separated by this extra step here in your mesophyll. So your catalase is not exposed to oxygen in the C4 plants, and instead you're delivering CO2 to the Calvin cycle. So everything's, hunky-dory there. The problem with this process is it requires an extra step, which is more energy inefficient. So if it's not super hot and dry, C3 is much more energy efficient, and the C3 plant will outcompete a C4 plant. But if it is hot and dry, C4 plants can function far more effectively and will outcompete a C3. Their example for this is like in early summer, you got tons of Kentucky bluegrass everywhere, but then when it gets hotter, crabgrass, which is your C4 plant, takes over. The other way of getting around this problem is with canned photosynthesis, and this is done by desert plants like cactus. And what they do is they do their light-dependent reactions, their carbon fixation. They do their light-dependent reactions during the day, and then at night they'll do their carbon fixation reactions to produce like their Calvin cycle to produce glucose. So In C4 plants, you're essentially segregating the light-dependent and light-independent reactions according to space, where they're physically at. And CAM plants are doing it at night, and so chronologically separating the two. And then C3 plants just do everything all together at the same time. If you have any questions, comments, concerns, let me know, and I'll see you next time.